Hi, in this video, I'm going to talk about co-immunoprecipitation. So co-immunoprecipitation is a very good technique to study in vivo protein-protein interaction. So inside a cell, if we suspect that two proteins, let's say protein A and protein B, interacts with each other, then we need to devise a technique and think of a technique by which we can show that protein A or protein B is really interacting. And for this, we have to do co-immunoprecipitation. Now, one thing we should keep in mind that for this co-immunoprecipitation, we should have antibody against both the proteins. If we don't have antibody against both the proteins, then this process is not possible. So for co-immunoprecipitation, we have a cell. So let's say inside that cell, protein A and protein B are interacting. We suspect that protein A and protein B are interacting. So what we would do, we would lyse the cell and put it in a buffer, and then we would add a bead, magnetic bead containing antibodies against protein A. So here our antibody is anti-A. So anti-A antibody is linked with these magnetic beads. So anti-A antibody, which are monoclonal antibodies, would bind with our protein A. And we don't even know that protein A is bound with protein B or protein A is not bound with protein B. Let's say, as this is a monoclonal antibody against protein A, we are sure that our magnetic bead containing this anti-A antibody would bind with protein A. Now what we would do, we would centrifuge this total solution, and after centrifugation we would get a fraction, that is pull down fraction. So this is a pull down fraction. So we expect that these magnetic bead along with this anti-A antibody would pull down A. So if protein B is interacting with protein B is interacting with protein A, then we would expect that when we are pulling down A with these antibody coated beads, B is also pulled down with the protein A. And if they are not interacting with each other, then we would find that B is not pulled down and B is not present in the pull down fraction. So in order to detect whether B is present in the pull down fraction or B is not present in the pull down fraction, we would proceed by these steps. So what we would do, we would collect this pull down fraction and we would process this pull down fraction and load it on a SDS page gel, a protein gel, and we would run the protein gel. So after running the protein gel, what we would do, we would perform a Western blot, and in this Western blot we would give the antibody against protein B. So basically we pull down A and now we are performing the Western blot using the antibody against B. And if in the Western blot, the diagram of the Western blot, we can show that in the pull down fraction B is present, then we can say A and B are interacting. So we have pulled down A here and we have loaded the pull down fraction here, and we expect that in the pull down fraction B is present. So B would be present in this particular gel, and B would be blotted in the PVDF membrane. So when we put antibody against B, we can see band against B. So these bands here, so these are the loading controls like beta actin band. So these bands represent B over here. So from this particular Western blot profile, we can say that our protein B is actually present in this pull down fraction. So that is how we can get an idea that whether two proteins are interacting inside a cell or not. So there could be two possibilities: either they are interacting, they are not interacting. So let's say we have a control actin band, beta actin band, And this beta actin band is a loading control, so it would be present in the either conditions, whether B is present or whether B is not present. So if B is present, we would get a positive band for B when we are blotting the gel. After running the pull down fraction, when we are blotting gel, we would get a positive band for B. And if B is not present in the pull down fraction, that means if we think that B is not actually interacting with A, then what would happen? There would be no band for B, but there would be the loading control, the control band would be there, the loading control band would be there, but there would be no band for B. So from this particular graph, we can say that these two proteins A and B are not interacting. Most likely they are not interacting. But from this graph, we can say that protein A and protein B are interacting because we have first pulled down A with a particularly monoclonal antibody coated bead, and then in that pull down fraction, we are able to show that B is present. So we can say that inside the cell, in vivo situation, A and B are interacting together. That is why we are seeing the band in the B. I hope you enjoyed this video and this video is useful to you. And if you like my video, please give it a quick thumbs up, and thank you. And don't forget to subscribe.